defect. So I was really drawn by this disease when I learned about really the catastrophic consequences of a complete deficiency. And to set the stage for the science, I thought I'd share with you two case studies. So this is sort of like medical grand rounds at the Broad. And one is about a case that I learned about through a collaborator of mine, Mark Batshaw, of neonatal onset. So it's a 36-hour-old male infant, was noted to show excessive crying and irritability, and was found to have an exceedingly high ammonia, 1,500. This child continued to progress, suffered a cardiac arrest, stabilized, and then sent to a tertiary care center for treatment, and had elevated ammonia, and then was diagnosed through analysis of plasma amino acids for having OTC deficiency. By the time the patient was diagnosed and stabilized, he had 3 days of hyperammonemia. Supportive care as hemodialysis and intravenous scavengers ultimately led to a reduction in ammonia to about 150, and the patient was eventually discharged, but the problem was with this persistent hyperammonemia for 3 to 4 days, had significant developmental delay, was encephalopathic by EEG, and showed brain atrophy, so had suffered irreversible damage. Went ahead with a liver transplant as a way of replacing this patient's liver OTC gene, but eventually succumbed to complications of the liver transplant. So the unmet need here, when I learned about this, is how to salvage function in the neonatal episode, which is the most severe, and how to replace the need for liver transplant. A partial defect in OTC deficiency can emerge at really almost any time, and this is an interesting case of an otherwise healthy young man in his late teens brought to the emergency room with confusion and lethargy. And at presentation, he had progressed with stuporous and declining function. He had slightly elevated liver enzymes, but his serum ammonia was very high. This was an otherwise healthy male. The history revealed that he was an athlete trying to gain acceptance in college, and had started protein supplements and was practicing for the basketball team. Metabolic workup was begun, unsure why his ammonia was so high. To combat the hyperammonemia, again, was hemodialyzed, was eventually controlled, but because of the persistent high ammonia, he developed CNS consequences, cerebral edema, and eventually expired and was determined to have OTC deficiency. So the unmet need here is, if diagnosed and if you survive, you have to prevent the repeat episodes of hyperammonemia. So I'm going to focus initially on what can we do with gene therapy or genome editing to help those patients with a partial defect that do not have an episode in the neonatal period, but at a subsequent time may have an episode that they're at a repeat risk of. And we took a standard gene therapy approach using a vector that was discovered in my lab called AAV8, which showed a high tropism for hepatocytes, as shown here in mice that were injected with some of the early generation AAVs and then AAV8. With this kind of gene transfer, we felt it may be possible to use this to treat patients that have liver metabolic disorders, because every time there's a transduction event, you could reconstitute the urea cycle. We're able to show that, that this kind of efficiency of gene transfer seemed to be relevant across multiple species, including primates, with one exception being dogs, in which the transduction was lower. And this was just due to some differences on the surface of the capsid that may render better tropism. So the initial application of AAV8 in the clinic was not in OTC deficiency. It was in another liver metabolic disease that's called hemophilia, which is due to a defect in a blood clotting factor that is produced in the liver. And so we, upon discovering AAV8, evaluated the potential of this vector for reconstituting factor IX expression in a dog model of hemophilia. The animal was initially infused with an AAV2 vector, because that's really all we had at the time. was able to show a low level of expression of factor IX in the blood, but was not therapeutic. And then subsequent to this, we discovered AV8 and injected AV8 into the same dog, and we were able to show much higher level of expression that was stable for the life of the dog. This was 12 years. And what's curious about this is AV genomes do not integrate. They persist extrachromosomally, with some exceptions that we'll talk about in a minute. But with these proof of concept studies in animals, large animals, a group of investigators proceeded with a clinical trial in patients with hemophilia B, infusing a vector into adults with factor IX deficiency, and showed an increase in factor IX activity over time that then persisted in some of these early treated individuals now for about 6 to 7 years. So when we finally, after decades of trying, generated a vector that we thought would have the efficiency that could be useful for liver gene therapy, we then, following this proof of concept, moved into OTC deficiency, but in this case, it was in patients that had a partial defect. So we had a mouse model, the sparse fur mouse that had a partial defect and did not have severe sequelae at birth, and eventually, though, was protein intolerant. So this was an experiment, published study, where we infused into these sparse fur ash mice that have a partial defect different doses of an AAV8 vector. And this is administered post-weaning, so they were adolescent in terms of development. And these are different doses of AV. And then we subsequently, a couple months later, evaluated their liver for OTC activity. Under untreated conditions, there's no activity. Wild type is here. And you can see here in a dose-dependent manner, we were able to achieve pretty efficient gene transfer, even down, and this is a fairly low dose at 10 to the 9th, quite a few cells that were expressing the OTC enzyme. And importantly, that this engraftment, as illustrated by a drop in urine orotate that's elevated in this disease, was stable for up to 40 weeks as long as the animals survived. And the animals became more protein tolerant. So we thought about what to do as a next step. And what we ended up doing is partnering with a local company, Dimension Therapeutics, that I had founded, and developing a phase 1 clinical trial for AV8 expressing OTC in a gene therapy approach in patients that have late onset OTC deficiency. This IND is open and should be enrolling this year, so I'm anxiously awaiting to see how this plays out. The therapeutic value here is that this could potentially prevent repeated episodes of hyperammonemia that, if not addressed early, as you saw in that second case study, could be lethal. But while I was doing this work, I kept thinking about this patient that I had seen when I was a medical student back at the University of Michigan. And this is, again, an incredibly tragic situation for young parents who have a normal born young boy, great APGAR scores, and then within 12, 24, 36 hours just basically decompensate, and there's 50% mortality. Again, the unmet needs here are to prevent loss of function and to affect survival in the neonatal episode, but then also to prevent the need for a liver transplant. So we asked the question, is the expression of AAV fast enough so that if we could administer a vector in a child who was just recently diagnosed, could it potentially manage the hyperammonemia fast enough for us to affect survival and protect the CNS? This is an experiment where we injected an AAV8 vector expressing OTC deficiency, and in this case it was the sparse fur ash mice because it's what we had at the time, and then harvested the liver at 24 and 48 hours and analyzed for expression of the OTC protein. And what we have here, this is wild type and control, 24 and 48 hours. Actually, we have pretty good expression at 24. This is a Western blot. This is background protein, 24, 48 hours here. We're achieving really stable, high-level expression within a day. And that would be a value in these patients because it takes a while to mobilize a team to hemodialyze and to provide this intensive therapy. So with that, we then did the experiment where we injected the vector in newborn SPF mice and then followed them over time to see how long that this would engraft, because we were worried that the vector expression would go down because the genome's not integrated. And when a newborn liver proliferates, it'll dilute out the genome. And so here's an experiment where we injected newborns with our AAV8 vector and analyzed the animals at a week, 4 weeks, 8 weeks, and 12 weeks later. And what you see is a pretty dramatic decline of expression due to the proliferation of the liver. Again, these are the controls. That correlate with here, if you look at efficacy, this is urine orotate that is elevated in the untreated animals. This is wild type, and this is a log plot. And at 4 weeks, we actually still have some level of protection, but by 8 weeks, it's gone. So we thought that this could be useful, potentially, for treating the neonates, the neonatal onset, but maybe not sufficiently efficient to prevent the need for liver transplant. And what's curious, as you'll see here, is there are some cells that persist. We didn't think this would be efficient enough to be effective, and this just relates to the integration of the genome into a proliferating cell type. You can actually see sort of clones. So we generated a knockout mouse in which it had no activity, and the phenotype of which is the animals die within 18 to 24 hours from hyperammonemia, an exact phenocopy of patients with a complete deficiency. And this is the survival of the untreated animals here in red. And then we injected the animals with an AAV8 vector at birth, and we were able to show protection that they didn't die, as shown here. But within 30 days, they eventually did. Is that expression waned due to the proliferation of the liver? So you can say, well, why don't you just inject the vector a second time, which we did in the mice, but eventually that waned. And then if you inject it twice, you can get survival. So you may say, well, just keep injecting your vector. But in humans, they develop antibodies against the vector, very high levels, which we do not see in newborn mice who are more tolerant. And so you really only have one shot with the vector, and that's at newborn. So while we may affect survival, they'll still need a liver transplant. So therein comes genome editing. Is there a way in which we could potentially drive the integration or correction of the gene in these newborn animals so that we have more efficient and stable reconstitution of OTC so that you could prevent the need for liver transplantation? Wasn't sure, didn't think that that would be fast enough, though, in order to treat the neonatal episode. So we've evaluated a number of different enzymes, and in the setting of AAV vectors, because we wanted to leverage our experience with AAV in the liver, and we're able to show in vitro that we could get reasonably good targeting, and in vivo, which I'll show you in a minute, also fairly efficient HDR. So just two vignettes about how this is played out. One's published and one isn't published. The first experiment was an attempt to try to correct the mutation in a sparse fur mouse, in which we injected vector both in newborns and in adults. And this is the Staph aureus version of Cas9 in one vector, and then the guide and a donor template and a second vector, injected into newborn mice, and then the animals were followed over time for correction of the mutation. So this is the molecular target here, in that the mutation is a G to A nucleotide change at the border of the 4th exon that affects splicing, so that only 5% of the transcripts are appropriately spliced. And so for our guide targets, we evaluated a number of targets, one of which was in exon 4, and 2 of which were in the downstream intron. Screened those in vitro, which showed targeting for RNA1 and RNA2. And then also in vitro, we're able to affect HDR correction of this point mutation at pretty high efficiency. So we decided to proceed with 1 rather than 2 because inadvertent insertion or deletion with indels without HDR we felt here is less likely to affect the residual function of the enzyme, whereas in the exon, whatever was left, if we introduced a break and NHEJ without HDR, we would decrease activity. So here are the results in which we injected the vector, these 2 vectors into the sparse for newborns, and we evaluated their livers at 3 weeks and 8 weeks later for expression of OTC enzyme. And you can see that there was some evidence for correction at 3 weeks, and it was higher at 8 weeks and then was stable. Our untargeted controls are always irrelevant guides, and everything else is there. And then this shows under high magnification these sort of tufts of expression. So what we've likely done is early on corrected a mutation, and then as the liver grew, we had clonal proliferation. You know, one other interesting thing about these experiments has to do with the notion that you don't need an endonuclease if you have AAV to get gene correction. And there's an emerging new data set. But this is our control for that, and we just don't see it with any efficiency. We've tried to achieve that. So those that are in the business of improving the technology of endonucleases, I think you're still going to have a job. This just shows the level of correction that we quantified here in terms of percent that a hepatocyte transduction by immunohistochemistry. So then we asked the question, is that level of gene correction sufficient to protect a partially deficient patient from a hyperammonemic crisis? And if you remember the case study, it was a young boy who had been well, had been growing, and was started on protein supplements to improve his athletic performance. And it was that protein overload that led to the hyperammonemic crisis that he had. So we simulate that in our mice because they're otherwise stable on their chow diet by providing them with a high protein diet at 7 weeks of age. So in these experiments, these are sparse for animals. We treat them as newborns, and then we start them on a high protein diet. And without treatment or the control experiment, they start to die during this period of time, and they develop an elevation in serum ammonia, as shown here. But in our treated animals, their ammonia is not normal, but it's improved, baseline ammonia, and we get complete survival. So this was sort of a proof of concept, and we kind of figured that the critical step of HDR would require intervention as a newborn because of the notion that proliferating cells are more conducive to HDR. But we went ahead, one other point has to do with the concern of the fact that we continue to express Cas9 in the guide with an AAV vector because it's not stable. But when you introduce the vector into newborns, you get dilution of the vector And that's the whole problem of why we're going to genome editing. And this is just an experiment where we followed the animals injected as newborns 1 week, 3 weeks, and 8 weeks later for expression of Cas9. And you see a loss of protein expression, so it's no longer detectable, a loss of genomes, and a loss of RNA. So introducing a non-integrating vector into proliferating cells to transiently express Cas9 may be one way of getting around the issue of ongoing expression of the Cas9. So onward to the adult experiment. We went ahead and just did the experiment where we attempted to inject the 2 vectors into adult SPF mice, in which, and then we followed them subsequent to that for several weeks. We didn't think this was really going to work that well, and it really didn't work that well because we required HDR to correct this mutation. But we could see in the treated that was not found in the untreated some evidence for some gene correction, and as under high magnification, you can see here the cells that we corrected and reconstituted OTCX expression. It's a very different pattern than the newborn. In the newborn, you'd see clones of cells, and these are just isolated cells. So we harvested tissues and analyzed them using molecular tools for the percent of indel formation and then the percent of HDR by correction. And we've analyzed neonates, analyzed 3 weeks or 8 weeks after the gene editing, and then adults, both a low dose and a high dose, several weeks after genome editing. And as others have seen, which is quite remarkable, is the efficiency of the break and the indels is really quite high in adults. That's not the rate-limiting step. In fact, we find it higher. We could probably even go down lower on our vector. But that the percent correction, which requires HDR, is substantially lower. So the breaks in the NHEJ works well, but HDR doesn't. We went ahead and continued to analyze those animals despite the fact we did not have much correction by HDR, assuming that when we got to the high protein diet that there would not be any protection. And so in this experiment, we had followed the percent survival over time in these adults that were treated. And we were waiting to put them on a high-protein diet, but we were surprised when Lily Wang, the chief scientist, came into my office and said, the animals that got the targeting construct are starting to die before we give them the high-protein diet. So we had to obviously try to understand the mechanism of this. They were losing ureagenesis, as evidenced by ammonia and the serum orotate. So one explanation is these are adult animals, and we've caused hepatitis because Cas9's a foreign protein. But when we looked at the livers and looked for evidence of hepatitis, there was none. So somehow, with a targeted construct and not with a control, we diminished the ureagenesis. And we went ahead and analyzed the indels, very high frequency indels, 44%. With respect to, this is specifically deletions and the size of the deletions. And we compared neonates to adults, neonates here and adults here. And most of the indels are really quite small, 0 to 10 base pairs. But there are some in which the indels are higher. And the real difference became evident in terms of large deletions. That the adults were predisposed to a much higher number of large deletions that when mapped actually overlap or delete a portion of exon 4. So these large deletions decreased the residual activity of the hypomorphic mutation in adults that we did not see in the newborns. And so what we think happened here is when you treat a hypomorph by genome editing, if you want to correct a mutation, you have to be careful that your intervention does not inadvertently decrease activity of the hypomorph. So this is schematically kind of how I view it simplistically, is that the sparse for ASH has residual activity compared to the wild type. When we genome edited the newborn, we were able to correct expression in some of the in some of the cells to wild type levels, but in the adults, the number of corrected cells were diminished, and we eliminated expression from many of the residual cells. So, so just going to finish with one last vignette, and this is addressing two issues. So what I just described may be useful in treating newborns to prevent the need for liver transplant. That's what we hope. But as designed, it's only specific for that mutation. So how can we make this more generic? And can we have, I call it a hit and run, or a, whatever you want to call it, a vector that can express high levels of OTC acutely as an AAV, and then integrate and provide long-term expression. So we can, with one intervention, conserve or preserve function as a newborn and achieve stable engraftment of the gene. So the approach that we took was not to correct the mutation here, but to design as the donor a functional mini-gene, promoter, wild-type OTC with flanking sequence, and that's what we did here. Now, you could drive this integration anywhere you wanted it to go, but just for simplicity's sake, we drove it back into the site where we had corrected the mutation. So that would be at the guide that's in that adjacent intron. So when we inject this vector, we have the Cas9 construct, and we have the donor construct with the guide, and this in and of itself would express OTC upon entering the cell. And then as the cells divide, hopefully integrate into the genome. So you have acute expression from the unintegrated AAV genome, and then long-term expression from a genome editing event. And so this is our data on the ability of this construct to achieve site-specific integration and long-term expression. So here's expression of OTC enzyme activity 3 weeks afterwards and 8 weeks afterwards. And you can see we get very high level of stable engraftment integration of the genome. Now, this is a complicated experiment because you have potential for expression of many different forms. It could be an integration event that is directed via a break at our site by HDR. It could be a non-homology directed integration event, or it could be residual unintegrated AAV genomes. But with the controls, with the untargeted, which would basically control for everything else but the targeted event, you can see a substantial improvement here to here. And here's the level of long-term OTC enzyme activity that we could reconstitute with this sort of hit-and-run approach. At 3 weeks, it's low in the untargeted, but very high in the targeted. And by 8 weeks, you basically eliminate any from untargeted events, and you have high level at 8 weeks. And the percent of OTC enzyme activity here is almost normal. So how well does this work in affecting protection long-term? And same experiment where we have newborns injected with 2 vectors. These are the sparse fur animals. 7 week, we put them on high-protein diet. We then follow survival and then follow ammonia. Untreated animals here, and you can see we've almost normalized the ammonia. This is actually more efficient than correcting a mutation, and we have survival as opposed to the controls where they all die. In terms of the actual trying to sort out how much of this is due to HDR at the target site versus other integration events. We've conducted a number of molecular studies, in particular here, using a more complicated protocol that I can talk about later if you'd like. But this is the level of HER-mediated gene editing that we're getting, which is really up to around 20, 30, 40%. The amount of editing that we get without the nuclease break is really decreased by about 50-fold. So how do we envision this from working? This is an experiment where we follow the level of OTC enzyme in the hepatocytes with genome editing alone, and you can see there's a delay, right? You don't see much at day 1 or day 3. This would not be sufficient to treat the hyperammonemic crisis. Gene therapy alone, you get great expression early on, and then you just get a residual amount later that's not necessarily enough. But the combination of the two, we do get expression early and we get expression late. So for this kind of unique set of genetic diseases, that provides an opportunity for us to exploit the role of the dividing cell to affect integration in HDR. I think there may be a real clinical value here. So for us, the next step before we would ever think about bringing this into the clinic is how well does this technology work in large animals? And that has been an important vetting step in gene therapy. So there isn't an OTC deficient large animal model, but we're just taking this stepwise and asking the question, can we achieve efficient NHEJ and then HDR in large animals? In our model system is a knockdown of a liver-specific gene, PCSK9, and we've just started this work in rhesus macaques with AAV, where we inject, in this case, Cas9 with a guide into adult macaques and then also newborn macaques, and then follow them over time for a decrease in PCSK9. And so we have evidence. We obtain the guides, and we've been able to show in vitro that we can achieve very high level of targeting. And we've now moved this into animals, and we're just in the process of working through monkeys. But we have an experimental system that we've developed to be able to study human targeting of human genes. And it's interesting. What we do is we put the target in the animal through an AAV, human PCSK9, and then we come back with AAV to try to target that to see if we could decrease expression in vivo assay. So what we're looking for is a decrease in PCSK9 expression, human PCSK9 expression, following the targeting constructs. And you can see untargeted, they stay high, and in a dose-dependent manner, we actually have a quantitative assay for showing targeting in vivo to a human transgene. So just want to end there and thank my collaborators both at Penn and at Children's National for working with us on this project that we've been working to try to affect this disease for almost 3 decades, and I think we may be on the cusp. So thank you very much. Time for a few questions. I think we're right on time. Yeah, please. Yeah.
So, I mean, there's thousands of these rare diseases out there. Just in terms of thinking about what are the, are there kind of common principles? Can you take what you've done for this specific disease and translate that to some of these other diseases? I mean, I think one of the hopes with genome editing technology is that it can be a generalizable approach.
All right. So I would say the one thing that we need to keep in mind as we move into the clinic is focus on diseases where there's significant unmet need, where the situation is dire. Because until we get into humans, we don't know what the risks are.
Yeah.
But this is a true platform. If we could figure out how to target a hepatocyte from a newborn adult, there are 4 or 5 urea cycle disorders, there are amino acidopathies, and with newborn screening, that may be the time to intervene rather than wait for symptoms to occur or the need for liver transplant. So So, the fact is, though, from our experience with HDR, it's much more efficient in newborns. Liver is easy to target. We just have to understand more the risks, both in large animals and then in carefully selected human trials where the situation would warrant this. But then this could be a way in which, with newborn screening for any liver metabolic disease, you get your vector when you're a kid, when you're an infant. That's the dream. Now, I know this is being taped, so let me just say that it's going to take a while for us to get there. Okay.
Thank you, Jim.
Okay.
Thank you so much, Wilson. Dr. Wilson, great talk. I'm Yichuan Wang from NCI Frederick. My question is, what could be the possible reason for the high correction rate in neonatal mice and the low correction rate in adult? So whether there is any possible strategy to improve the correction rate in adult organ or even animals? Thank you.
Yeah, the question was the difference in HDR in newborns versus adults, and if we understood it, then we can maybe design a way to improve it in adults. I think we should talk to our colleagues who know a lot more about DNA repair than I do, because it has a lot to do with the cell rather than the construct itself. But that's that I think is an incredibly important question that I don't have a solution for at this point.
Question here.
Yeah.
Two questions. One is, in your first experiment where you're using the 1.2 kb donor, how far was the mutation from the guide target site? So you had it was in the intron.
Yeah, it was maybe 30, 40 nucleotides, something like that.
So
yeah.
pretty close.
Yeah,
so
it
the
was
second
close.
question, did you, you mentioned that you had high HDR in the second experiment when you used the mini gene. Was the HDR higher than in the first experiment when you were using the sort of standard donor?
The HDR, as measured by enzyme activity, was higher. It's an interesting point. In terms of molecular characterization, probably not. I think I haven't actually thought about that, so thanks for the question. But there's regionalization of OTC activity in the liver, and when you correct it, it only reconstitutes expression where it was normally expressed. When we put a constitutive promoter, we're getting expression in all hepatocytes. That may be one explanation.
Okay. In the mini gene, what promoter were you using in that case?
I think that's our liver-specific promoter, TBG, that we use.
Okay, thanks.
This hypermorph being converted to a loss of function phenotype, that sounds a little alarming. Is this been observed in other situations, or what are your thoughts on all the clinical ramifications?
Right. Well, I think we should go into nulls first so we don't have to worry about it, but that's dodging the question, right? I think it's an issue with sickle cell, because you convert a sickle mutation into a thal, and I think they're struggling with that from what I understand. It's a pretty important application.
Thank you.
Sure.
Hi everyone, my name is Salil. I'm a postdoc in the lab.
tell us about in vivo genome editing via targeted integration.
Okay, thank you for introduction. First, I would like to thank the organizer for giving me such a great opportunity. So today, I'll talk about non-homogeneous engineering mediated targeted integration for in vivo application. Okay, so as you may know, 2 DNA double-strand break repair machinery is important for targeted genome editing. Okay, so if NHEJ work to repair the double-strand CRISPR-Cas9 induced double-strand break, so they sometimes introduce cannot see pointer. So cannot see pointer. Maybe my back is white. Sorry. Okay. So anyway, so NHEJ introduce insertion and deletion. Then we can knock out the target gene with random mutation. In contrast, homologous recombination can introduce any sequencing at target sites, so which enable us to do gene knock-in as well as gene correction. So however, these 2 DNA double-strand break repair machinery are controlled by cell cycles. Although NHEJ works through all of the cell cycles, homologous recombination HDR only work S to G2 phase. So that's why current HDR-mediated targeted gene knock-in is only achieved in dividing cell, but not non-dividing cell. So because most of the cell in our body are differentiated post-mitotic cell or quiescent stem cell, so current HDR-mediated gene knock-in is still inefficient for in vivo application, except some dividing cells such as liver. So that's to overcome these hurdles and enable gene knock-in for all of the tissues. So we propose to hijack highly efficient non-homogeneous engineering system for in vivo. First, so we compare the existing targeted gene knock-in method. We constructed a series of donors for HDR and HEJ or MMEJ mediated target integration. So MMEJ is a microhomology mediated engineering, which needs a microhomology to target the integration site. And this is another minor DNA repair machinery. The NHEJ-mediated targeted integration was initially reported with zinc finger nuclease 2013. So we optimized this system for CRISPR-Cas9. The trick is very simple. We insert Cas9 guide RNA target site on 1 or 2 guide RNA target site on donor. In the cell, this donor DNA will cut by Cas9 and insert at target site. So we measure the DNA targeted gene knock efficiency by GFP or MCHERI expression. Then here is the result in dividing HEC293 cells. So as you can see, targeted gene knock efficiency of NHEJ-mediated method is 10 times higher than other homology-dependent method. So that's why we named this method as homology-independent targeted integration, HITI. So this animation shows how NHEJ work for targeted genome editing. So we choose most popular SPCas9, which target 20 base pair specific genomic sequence and 3 base pair palm sequence. And SPCas9 introduce double-strand break between position 70 and 80 and generate a brand end for most of the case. Once CRISPR-Cas9 cuts the target site, NHEJ component will access to the double-strand break immediately. Then repaired, most of the case, the repair is error-free, is no mutation, which we call error-free. then CRISPR-Cas9 target site reappear, then cut again and repair again. So this cycle should be supposed to repeat many times in the cells. But eventually, NHEJ introduces insertion deletion. Then Cas9 recognition site will disappear, then cannot cut, then this mutation will fix at target site. So that's why we can easily knock out the target site with random mutation. But regarding to HICHI, so we introduce donor DNA, which we want to insert at target site. And we insert exactly same 23 base pair Cas9 target site to the donor on the donor. But we put with opposite direction between donor chromosome and donor. Then in the cell, Cas9 cut both donor and chromosome. The NHEJ component will accumulate soon. If this donor will integrate with desired direction, then after NHEJ mediated ligation, so Cas9 cutting site will disappear for both sides. Then this donor DNA will integrate with desired direction and fix it. However, if this donor DNA will integrate with reverse direction, because most of the NHEJ is error-free machinery. Then after error-free NHEJ, Cas9 cutting site will reappear again. Then Cas9 cut it out this opposite direction of the donor. Then eventually this cassette will integrate with desired direction or cut it out. So this is expected molecular machinery of HICHI. Yeah, actually, the efficiency of reverse direction is very low, so which suggests HICHI is a unidirectional targeted integration method. And also 90% of the junction site sequence of the correct clone doesn't have any insertion or deletion. So which suggests error-free NHEJ is a predominant manner for HICHI. Then we try to apply HICHI for non-dividing cell, first in vitro. So we transfected cultured or primary mass neuron cell with HICHI component to design knock-in GFP as a neuron-specific, the downstream of neuron-specific TUBB3 gene. And correct knock-in should result in the expression of fusion protein of TUBB3 and GFP, and the GFP should see the cytoplasmic localization. As expected, we can see the GFP knock-in with cytoplasmic localization, which suggests a correct knock-in, and HICHI can achieved even in non-dividing cell. And also we compared HICHI efficiency with other homology-dependent machinery, and only HICHI can integrate with higher efficiency for non-dividing cell. Then next, so we applied HICHI for in vivo. But to apply HICHI for living animals, so we constructed two AAV components and packaged into AAV cell type 8 or 9. And one AAV expression Cas9, another AAV express Harvard guide RNA and donor sequence. And in the co-infected cells, this donor will cut it out and target at the chromosomal target site. Then first we injected this HICHI AAV in the visual cortex of adult mouse brain, which is non-dividing tissue. And we can see the targeted GFP targeting knock-in at the downstream of TUBB3 gene. And the efficiency was 3% per cells and 10% per infected cell. And also we locally injected HICHI via intramuscular. Then we can see the GFP knock-in in skeletal muscle as well. We also tried systemic genome editing to this purpose, we choose AI-14 mice, which have a constitutive active strong CAG promoter at loci 26 loci. So we constructed AAV vector which can insert GFP as downstream of the CAG locus, and we can see the knock-in as a GFP positive signal in whole body. So to deliver this system, we inject AAV at P1 mice via intravenous injection. Then 2 weeks later, we could see GFP knock-in for many organs, but in particular, heart, liver, and muscle are a major target of them, and their efficiency was 3 to 10% per cell. And also, this knock-in efficiency is also depends on the titer of AAV. With high titer AAV injection, we can see up to 16% or 25% of knock-in for heart and muscles. Then to apply this system for future gene therapy, so we try to correct the inherited mutation using retinitis pigmentosa rat models. Retinitis pigmentosa, RP, is an inherited retinal degeneration disease and a common cause of the blindness in humans. So this rat model called as a RCS rat have a 2 kb deletion, including exon 2 at MERTK gene. So we constructed AAV which can insert exon 2 in intron 1 of this gene. We injected AAV by subretinal injection by locally at 3 weeks old and analyzed 1 month later. And correct knock-in and rescue of the expression level MERTK gene were confirmed by DNA level and mRNA level and protein levels. And also mRNA level shows each treatment can rescue by 4% of normal level, which was 0% in untreated rat. HE staining also shows a partial rescue of the thickness of ONL layer of RP mice, rat. And the visual function test via electroretinography also shows a partial improvement with HITI. So this is another one of the functional experiments. As you can see, healthy mice can follow the rotation of the strip. However, RP model rat cannot follow it due to the blindness. In contrast, the treated rat partially follows the rotation of the strip, and once the rotation starts to reverse direction, then they can follow a little bit. So these results suggest that which can correct the mutation and can see the therapeutic effect for visual function, especially for deletion type of mutation. Then in the last few minutes, so I'll talk about the more versatile hitch strategy to cure any type of mutation, including point mutation. So to this end, so we choose a progeria mice model. These have a autosomal dominant point mutation in LMNA gene, and this mouse shows a premature aging phenotype in large organ and show life short span. So to correct mutation for whole body, so we inject AAV with IV injection at P1 mice. Then we analyze. And actually, so the previous our hitch method can insert gene at targeted locus, but we cannot remove the mutation. But we propose a new novel strategy which can insert functional exon and 3' in front of mutated exon. Because this progeny mice model have a C2T point mutation at exon 11. So we insert exon 11, 12, and 3' UTR in intron 10. Then after integration, this integrated cassette should transcribe rather than the mutated exon. This mutated exon should still there. It won't affect the expression of the corrected mRNA. Then 7 weeks afterward, we confirm the correct gene knock-in with DNA or RNA level. And this rat shows a small body weight and short lifespan. And for both of the phenotype, is rescued by a Hitch significantly. So these results suggest that Hitch can cure even autosomal dominant mutation as well as point mutation. So this is the main collaborator of this project. Mainly this work we have done in Juan Carlos Belmonte Lab, Salk Institute. So I would like to thank for all of them. Thank you for the attention. Thanks for your attention.
I was curious, in your last experiment, did you look at the junction sites to see how much insertion deletions that you had?
Last experiment, actually we didn't see because we target intron. We don't care the deletion so much. But in the previous experiment for GFP knock-in, so we checked both targeted in there and knock-in. So actually, its efficiency is almost similar. It's deletion and knock-in efficiency was similar efficiency, 35%.
Thank you. Thank you for the presentation. I'm here. I have a question about in vivo mouse brain genome editing part. Let's say if you do delivery with AAV viruses and try to knock out genes in vivo, which are based on NHEG, those are quite high percentage, like, I don't know, 60, 80 people have shown. And your HITI is pretty much based on NHEG, and it was showing very high percentage in vitro. But what you got in vivo, it was strikingly low for me, like 10%. Do you have any explanation for that?
Actually, our HITI method is compete with error-prone NHEG. So error-prone NHEG introduce insertion deletion at target site, but Hitch inhibits this. So if we insert Hitch component, the expression level of, I don't know, is insertion deletion efficiency will be decreased. So that's why I think the insertion deletion will inhibit by Hitch, and also in our experience, the NHG activity, error-prone NHG activity is totally depends on the organs.
Okay, thank you, Kay.
Okay, thank you.
And our last speaker of the session is Heidi Williams from
It's my pleasure to introduce our next session, SynBio and Biotechnology, in which we'll hear from 3 researchers who are delving into molecular technologies for delivering, creating, and manipulating genes, respectively. Our first speaker is Luk Vandenberghe from the Harvard Medical School. Dr. Vandenberghe will start us off with a discussion of lessons learned in building AAVs.
Thank you. And while we're getting set up here, it's a tough presentation to follow, and I share your pain, and I think it's a very good thing to be reminded that the tools that we work with, which we all realize are so incredibly powerful, but applied in the appropriate way could lead to actually making a difference. I think that perspective is something that's healthy to be seen. And it's something that we in the lab actually pursue as well. So I'll be talking to you about delivery and delivery in genome editing, particularly around the adeno-associated viral platform, which you've heard a lot about this morning. I have a number of disclosures to make related to the science that we've done over the past decades. But as a reminder, and maybe as a segue to the previous presentation, we are very much in the science of trying to build therapies, and we do that at various stages, from the fundamental to the applied, and breaking down what a therapy ultimately looks like and how it's born out of the science that we're all pursuing. Maybe in somewhat of a simple way, we look at things as targets that are relevant to a particular disease process, as a molecular intervention that hopefully can interfere at some level with that disease process. And then what is often forgotten is that there's a delivery component that is key to actually making this combination of molecular intervention to the target be both safe and efficacious. And just to bring this to practice, what we mean with this is that targets can be genes, proteins, pathways, but also include a particular cell type and tissue that needs to be targeted. And the molecular interventions that are out there for a field such as ours, which is gene therapy or genome editing therapy, can be cDNAs, sRNAs, link RNAs, genome editing, and I'm sure I missed some species in there. And over the past 3 or 4 decades that really this field of gene delivery has emerged, people have evaluated a variety of platforms, whether it's non-viral platforms, but there's a field that has been strong for that entire period based on viral systems, and people have looked at adenoviruses, herpes viruses, obviously retro and lentiviruses. And I'll be specifically talking about AAV, which received quite a bit of attention this morning, and I think for the right reasons. But we'll be talking about some of the limitations of this platform and ways on how to improve it. It's a brief introduction. AAV is relevant for a reason to this application of genome editing, mainly that as a wild-type virus, when it was isolated, really it has a number of features that are quite interesting. For one, actually, it's a single-stranded DNA virus, and you probably are aware that as a homology repair template, single-stranded DNA may actually provide a benefit. Kind of out of the box, it has not been associated with any disease and therefore is labeled, at least for now, as non-pathogenic. It has a broad tropism, which allows it to target really a whole variety of tissues, although that's a trade-off towards specificities, which is really limited with this platform. One of the other downsides is that it seems to be endemic in humans, and I'll come back to that in a second. Now, once we eliminate all the viral genes from this viral genome cassette, which is flanked by these inverted terminal repeats, we can actually put in a therapeutic gene target, and that eliminates all these viral gene products, which really builds on top of another set of advantages of the system, leading to long-term stable expression of this platform that is devoid of viral genes, has a low immunogenicity, and really since the mid-90s, this has now entered in numerous, obviously, preclinical studies in people's labs, but has actually been in the clinic for now over 137 times, but really overall a very favorable safety profile. So there's clearly benefits. There's clinically validated targeting that shows both safety and efficacy, and I think those are the benefits that this field of genome editing wants to leverage. And on top of it, like I mentioned, it has a broad array of targets for in vivo applications. This is direct delivery in vivo. Whether it's the retina, we've heard about this from Editas this morning. The cochlea is some work that we've done recently. Liver, muscle, CNS, and that's clearly skipping a few targets. Now, there are clear limitations, and I made those specific here for the genome editing world. One is shared for the entire AAV gene therapy space, which is it's kind of the dirty little secret of this field. We can develop incredibly powerful gene transfer reagents that possibly, and there's emerging data coming out of clinical trials, could be truly incredibly therapeutic for patients. However, we believe that 30 to 90% of individuals, and those estimates obviously vary quite widely, may be ineligible of benefiting from these treatments based on pre-existing immunities. Pre-existing immunity is really an artifact or a consequence of the fact that many of us have been exposed in childhood to a natural AAV infection, which had no consequences for you except for the fact that it may prevent you from getting an AAV gene therapy later in life. The genome size is limited, and that's the reason why we work with dual vector system, and we have to be creative to kind of squeeze everything in between that limit. And it's really a fairly absolute limit. I run a research vector core that gets requests continuously from people trying to fit in 5.2, and really the deficit becomes quite obvious very quickly once you start exceeding this genome size. Now, AAV has been developed for long-term stable expression in often single gene disorders, and this has been a field that has been active for now 30 years. Now, all of a sudden, in this modality, long-term stable expression of, say, Cas9 is obviously something that is not wanted. So for us to leverage these benefits, we kind of have to take in these limitations. And then there's a very practical concern that for many of these applications, including the ones we've heard this morning, we need large amounts of AAV, and production, just making enough to actually bring this to the clinic, is often limited. And I'll talk about some of these limitations and how we are trying to address these from both a discovery as well as a fundamental perspective. So going towards mechanism, this is the transcriptional map and the genome map of AAV, and mapping kind of these limitations in red as well as these benefits on that genome. You can see that while for some of them they're really undefined where the mechanism lies, say for example for what gets us to a stable expression of this platform. For others, we quite clearly know that, for example, the capsid is associated with many of these phenotypes that we're interested in modulating. So the majority of what I'll talk about is exactly this viral capsids. These are the shell proteins that basically encapsulate the viral DNA. And this is, I'm not the only one in my field. There's an active set of studies that really tries to discover new phenotypes of different AAVs, which are either naturally occurring, and this is actually work that I did when I was in Jim Wilson's lab, whom you've heard earlier today, or by a variety of directed evolution methods or more rational design type methods. This discovery in general can be characterized by the fact that people try to identify winners in one or more selective criteria, say one of those limitations that I outlined before. And the problem often is that while we may optimize one property of delivery, it often comes at the deficit of another one. And really, the challenge has become to disentangle these different phenotypes. While you may optimize, say, tropism for your particular target cell, you may also want to make sure that you can still package your viral particle as efficiently as your original product. So really, in kind of summary, what we're trying to do often is study or modulate a particular property of interest while retaining the structural integrity of that particle, and obviously retain the desirable biology that we're leveraging for these clinical applications. And the challenge really comes in that these are all tightly linked in this viral architecture. This viral architecture I often compare with a Rubik's Cube. While you can try to align orange fields on one side, and that may be quite successful, as you all know, it's very easy to then at the same time disrupt the green fields on the other side. So we brought in kind of maybe a fairly naive idea that in order to get around to this, the constraints of this architecture, which really connects all these phenotypes, is there a possibility that we can somehow replay evolution? Because evolution at some level has been challenged by many of these same constraints that we're trying to optimize, whether it's assembly relevant to AAV production, whether it's immunology in terms of host response, or particular transduction efficiency of a target cell X or Y. So if we're able to replay this evolutionary path and B, understand what the selective pressures are at play, we may be able to learn some of the mechanisms behind these features. And our initial approach to achieving this was to use a method called ancestral sequence reconstruction, which is something that we haven't pioneered, but Joe Thornton, mainly out of the University of Washington, now in Chicago, has developed basically a computational method, just really briefly, uses sequence information that is available of contemporary species, integrates those in a phylogeny, and then really tries to reconstruct the putative sequences at these nodes. So this is what we did initially. We picked ANC83 here and ANC80. These are arbitrary names for these nodes. And while we were skeptical that this was going to work, our first result was actually, to us, a success, which meant that after production of one of these synthesized putative sequences, brought them into a functional context and tried to just get a viral infectious hit to work. What we show here is per microscopic field, roughly, we got one single transducing unit, as illustrated here by this GFP cell. And again, we felt that this was a heavily modeled approach. We knew about the constraints of this architecture. We thought this was very unlikely to work. Obviously, this isn't the greatest transduction efficiency, and what we did next, and actually we did it in parallel, was hedge our bets. And we worked, given the fact that this is a largely statistical process, we used maximum likelihood heuristics. We actually approached this by building libraries around these nodes, which would encompass basically all possible permutations around the sequence space around that node, informed to us by, again, this maximum likelihood prediction. And when walking through these fairly contained libraries, we stumbled upon a number of more productive viruses that were also more infectious. And this really was, at some level, a validation of our approach that while this may not have been truly ancestral sequences, they led us to functional intermediates of this fairly complex architectural space. So Eric Zinn in the lab really pioneered this work. Kind of as a further validation, we obviously knew that we produced something that was infectious. What we followed on is that these viruses actually produce analogous to AV2. Biochemically and structurally, they really looked very similar, and we were able to resolve them under electron microscopies. When we look at the sequence space here, these viruses are actually quite distinct, roughly in the range of 9 to 40% distinct from any known sequence in this space. And again, while building these synthetic particles, we still retain both function and structure. A further surprise was that when we put these to the in vivo gene transfer tests, that both in the liver, as well as in the muscle, as well as in the retina, this is all in mice, these viruses performed at least equivalent to some of the better AAV gene transfer tools, such as AV8, discussed earlier this morning. So we looked at this in liver. We brought this up to large animals, which is, as mentioned this morning again, is kind of the vetting prior to developing this to the clinic. And again, we show largely equivalence compared to some of these viruses. One of the applications that we were particularly interested in is a space where targeting and tropism was a limiting factor, and this is the cochlea, mainly targeting these cell types right here, which are dimly green, but actually dimly green. Most of these cells are targeted. These are outer hair cells These are the cells that are responsible for complex hearing, say at a reception or in a subway. And most of the technologies are unable to target these cells. Somehow we were fortunate that this newly developed synthetic capsid is exceptionally potent for these cells. Now, we wanted to really, this was all great, and there seems to be applications with the technology we developed, but we really wanted to move further in terms of mapping some of these phenotypes to this capsid structure. This is this is 1/20th of a particle. This is styled 20 times to make a full buckyball. And what we now can do with this ancestral sequence reconstruction methodology is really walk back through evolutionary times, if you will, or at least across different functional intermediates, because all these red dots have now been resolved. And each time, as you can see, there's only a minimal set of changes that is introduced, allowing us now to study the phenotypes at each of these nodes and map those back to function. One of the first observations was that the production quality of these viruses increased along one of these lineages, actually the lineage I just walked through, which led us to study the function of a cofactor that is involved in the assembly process that really glues together all these different tiles that I just illustrated. And one of the observations there is that this cofactor seems to be absolutely required in some of the branches. Some of these lineages have a significant shift in terms of how they utilize this assembly-associated protein called AAPs. Because of this quick divergence at this particular node, we were able to identify residues that were conserved in one branch on a consistent basis versus the other branch where they were divergent. Mapping those to about 10 sites led us to a minimal motif that basically optimized this particular assembly function for this particular set of AAVs, which we could transfer from an AAP independent or dependent virus, ANK82, into an independent virus. Now, switching gears, and I think I have 2 or 3 more slides. One of the real problems, as I mentioned, kind of this Achilles heel of the AAV gene transfer world is pre-existing immunity. And this is prevalent and lasting immunity following a natural infection of this endemic virus. Leads to neutralizing antibodies, which are highly cross-reactive with all the tools we use, and really prevents many of us to enroll in trials of otherwise perfectly fine future drugs. And then there's another issue regarding T-cell responses, which may impact the safety of some of these. And really our approach here is to avoid this historical irony by the fact that we reached out to nature to pick up those prevalent viruses, repurpose them for therapeutic applications, to then uncover that these prevalent viruses also were prevalent in our future patient population. So our goal here really is, and this, I think what I've shown you is one way of trying to diverge ourselves as far as possible from the naturally occurring diversities to go into a synthetic diversity. And exactly, if you look at the surface properties of ANK80, the virus I mainly described, this is really now 25% to 30% divergence. And this is exactly where most of these neutralizing antibodies will attack this particle. What it also allows us is now to fine map where these epitopes bind, or where these antibodies bind, and where these epitopes lie, by really, again, walking back these phylogenies or these lineages. So to close off, the field kind of has historically really elaborated and gotten quite far by taking viruses out of their natural environment, repurpose them in the lab for developing potentially transformative medicine, and we're seeing that right now in dozens of trials where really incredible results are being led, including some of the studies that were presented this morning. But there's a next version that I think is needed to really address in a broader way some of the delivery issues that you, as well as the field of gene therapy, have. And there we basically try to integrate the experience that we've had with smart synthetic solutions to, in our view, ultimately build synthetic viruses towards these goals. Just want to close off and thank all the people in my lab, Eric, who's been mainly involved in the ancestral sequence reconstruction stories, as well as many collaborators in the field. And anybody who may need an AAV vector could also is welcome to use our core referenced here. So I have time for a few questions if you have them. Thank you. Great.
Thank you very much. All right, our next speaker is Kai-Hong Wang, who is a postdoc in the Chin Lab at the University of Cambridge. Dr. Wang will discuss the Chin Lab's pioneering techniques in de novo genome synthesis.
Thank you for the kind introduction, and really thank you, Mr. Jafar, for being here with us, reminding us why we do science. Just try to make the world a better place for everybody. And the remote control. Okay, sorry. Right. Sorry, the beam is a little bit dim, but it should be all right. So basically, what I have been trying to do is basically following a seemingly delusive dream. Basically, if we look back into history, one thing has been said since the very beginning of life, and that is the central dogma, in which the DNA makes RNA makes protein. So the genome not only contains all the DNA, which contains information of life, it also contains a defined set of decoding rules dictating which one of the 64 triplet codons encode which one of the 20 natural amino acids. And here are the 20 natural amino acids. They do offer diverse chemical properties, but if you compare the chemical properties offered by the 20 natural amino acids compared to the unlimited chemical space in the universe, this is only a tiny speck in the unlimited, basically the sea of chemical space. And the idea is, if we could potentially go beyond the limitation of the 20 natural side chains on the 20 natural amino acids, then basically where evolution can take us, or more importantly, where bioengineering driven by human determination can actually bring us to. And because of the decoding rules are embedded in the very center of the whole genome, it is basically impossible to change the decoding rules to basically go beyond the 20 natural amino acids without changing the genome. And basically here, hence our idea to basically, if we could, de novo synthesize a brand new genome, then in that brand new genome, we can arbitrarily redefine a new set of artificial synthetic decoding rules, which potentially will enable us to venture beyond the limitation of the 20 natural side chains, which can bring us potentially many unlimited potential applications to better human life. And now the fundamental challenge is how we actually make a new genome, and how we engineer a new genome to actually go beyond the limitation of nature. If we look into the natural decoding rules, as in the wild-type decoding box, essentially there are tremendous redundancies how nature uses 64 triplet codons to encode only 20 natural amino acids. For example, in the serine decoding boxes, nature uses up to 6 serine codons to encode for the same serine and serine natural amino acid. And the idea we want to do is basically potentially, if we could reduce this redundancy and free up some of the natural triplet decoding space, then potentially we could use this freed up triplet decoding space to reassign to encode for unnatural amino acids with 100% efficiencies. However, because of the density of the target codons, TCG and TCA, in order to be reassigned to encode unnatural amino acids, it needs to be removed from the wild-type genome completely. And because of the number of TCG and TCAs, and because the density of TCG TCAs across the whole genome, the only realistic way of actually achieve such reassignment to encode for unnatural amino acids is to de novo make a brand new genome. Then the question is transferred into how can we actually find a method to allow us to actually de novo synthesize a new genome. Conceptually, there are two possibilities. The first one is the one pioneered by Gibson, by Dr. Daniel Gibson and other research pioneers in the Craig Venter Institute, in which they de novo synthesize a brand new genome and use that to replace the wild type genome in a cell, basically through the genome transplantation pathway. However, this so far has only been realized in the mycoplasma, which has one of the smallest genome in the entire life systems. The smallest mycoplasma is of the size around 600 kbs, and which is around 1/8 the size of E. coli genome. And basically, because of the compact size of the mycoplasma genome, makes that ideal system for the genome transplantation. But because of essentially it's tremendously difficult to handle a bigger piece of the genome, this method is tremendously limited to mycoplasma so far, and tremendously difficult to transfer to other domains of life, for example, into E. coli. And also there's another fundamental challenge with the genome transplantation, because we are really in the very early days of designing new genomes, and we are bound to make mistakes. And it has been shown that a single mistake in the synthetic genome is sufficient to derail the entire operation, such that there's no survival clones. Then basically we would only know that synthetic genome is not viable. There's no clue, there's no feedback to allow us to figure out exactly where the synthetic lethal is, where the design flaw is. And basically in order to answer all these challenges, we would like to entertain the second possibility of genome synthesis, which is rather than replace the entire genome at one go, we would like to basically replace a significant chunk of the genome at a time. Then basically, if we could iterate this approach, then basically in a few steps, potentially we could convert the entire wild-type genome into a brand new synthetic genome. And also, as we basically making the wild-type genome into a progressively more and more synthetic genomes, on each steps, we would like to have the ability to really pinpoint and identify potential design flaws, and with the ability to actually fix such design flaws before we move on to replacing the next junction of the wild-type genome. And potentially, if we can do this, and if we can really also do this with significant length on the walking along the wild-type genome, then potentially in a limited number of steps, we can basically make the wild-type genome into a brand new synthetic genome. Then the fundamental question is, can we actually do this? Right? Basically, the classical method to actually introducing a synthetic piece of DNA into the genome is by the classical lambda-red recombination in bacteria. And basically, you have one piece of linear double-strand DNA probably made by PCR, and you transform that double-strand linear synthetic DNA, which is coupled with a positive selection marker, into the cell. And at the same time, you basically do recombination. And you select for the right recombinant by selecting the coupled positive selection marker. And this has been essentially the default tool for introducing synthetic piece of DNA into E. coli genome for decades. However, it does come with several limitations. First, the efficiency of the overall recombination is really, really low. And that is fundamentally because it is a coupled event between the transformation and the recombination. Essentially, both of the two events are really, really unlikely. And for the two to happen at the same time becomes extremely unlikely, which basically fundamentally makes the efficiency, overall efficiency of the whole process very, very low. You typically have maybe around like a few hundred clones per experiment. And also making things worse, as the size of the synthetic DNA gets longer and longer, the efficiency of the transformation drops down really, really fast. And such that beyond, say, a 10 kb of the synthetic DNA, it's impossible to get any clones at all. And also, the integration of the synthetic DNA into the genome by the classical recombination is purely a function of the homology regions, which is not 100%. You routinely have off-target integrations onto alternative loci of the genome, leading to false positives. Right? Essentially, the typical hit rate into the right location by the classical recombination range from like a few percent to maybe 80–90%. And in this particular case, it's around 50%. And we really need to address all these fundamental limitations before we can really have a reliable method of walk along the genome and replacing the wild type one into a synthetic one. And how do we actually answer like these limitations, right? And basically, in order to do that, we designed and developed this new platform, which we call replicon excision enhanced recombination, or REXR. And the principle is really simple, right? The fundamental reason we have the limited efficiency and the length dependency in the classical recombination is because the transformation and recombination is coupled. Then basically the key in solving these limitations indeed lies in decoupling the two coupled events. Right? The way we actually decouple the two is we actually, rather than transforming the synthetic DNA directly into the cell, we actually first put the linear synthetic DNA onto an artificial replicon, in this case, a bacteria artificial chromosome. And we transform this bacteria artificial chromosome, which holds the synthetic DNA, stably into the cell. We pick a colony, grow it up. Then in that population, every single cell will contain the back, which contains the synthetic DNA. Right? Then basically, we use CRISPR and Cas9 and excise the synthetic DNA from the back cassette, from the back carrier, exposing double-strand breaks there and there in the REXR2 cuts. And also in a similar REXR4 cuts, we not only excise the synthetic DNA from the genome, we also basically make a double-strand breaks on the genome at the designed locus. And finally, basically, we basically select for the right integration by essentially select for the gain of the coupled double selection cassette together with the synthetic DNA, and also the loss of a different double selection cassette, which we use to mark the designed integration site on the genome. And in this case, by selecting the gain of the +2, which is coupled with the synthetic DNA, and the loss of the negative selection marker -1, which is used to mark the genomic locus of interest, essentially by gain of +2 and loss of -1 at the same time. This allows us to have 100% locus specificity. And for the REXR2 and REXR4, because of the decoupling of the transformation from the recombination, essentially we achieve tens of thousands of efficiencies with REXR2, and actually tens of millions clones per experiment with REXR4. And we have the same efficiency regardless whether we are integrating a 2 kbps or whether we are integrating a 200 piece case, 200 kbps. Then basically we first try to use the REXR strategy to actually integrate long piece of synthetic DNA as insertions. In this, basically what we do, we first mark the genomic locus of the interest for the integration with the insertion of the first double selection cassette -1+1. Then we synthesize a 90 kb synthetic piece of DNA and put a second double selection cassette -2+2 onto it. And also we put like 5 different genes from the luciferase operon, which basically, when all present in the same cell, will give the cell bioluminescence. Basically, we spread the 5 luciferase genes across the 90 kb synthetic DNA as markers to make sure, essentially, if we have a cell that actually lights up, that indicates has all the 5 marker genes. Then basically, we cut out the synthetic piece from the back, or we cut out the synthetic piece from the back and also excise the double selection cassette from the genome. Then basically, if we just cut out the synthetic piece from the back, following the REXR2 strategy, then essentially the synthetic DNA will integrate into the genome through the strand invasion pathways. But if we basically cut both the back and the genome, then basically we actually generate 4 double-strand breaks. Then in this case, the synthetic DNA can actually integrate into the genome through the strand annealing pathways. And the strand annealing systems is basically fundamentally a lot more efficient than the strand invasion. And then we select for the gain of +2 and loss of -1. And basically we have the right integration of the synthetic 90 kb of DNA integrated into the right locus. And if any one of the marker genes is missing, then the cell will not light up. And in order for the cell to light up, you need all the 5 genes to be there. And basically, with Rexer-4, we really have like tens of millions of clones per experiment. This is a 10,000-fold dilution plate of one Rexer experiment. And basically, if you turn off the light, then basically you see every single cell on the plate will light up, indicating it has the entire 90 kb synthetic regions, synthetic DNA integrated into the right locus. And if you superimpose the two pictures, really indicates we have 100% integration efficiency into the right locus. Right, and that is for insertion. Then basically we can apply the same principle for replacement. And in this case, essentially you just put the 100 kb designed to replace the first 100 kb of the wild-type genomic sequence with synthetic sequence. Then you do the Rexer experiment. Select for the gain of +2, loss of -1. Then you have your first 100 kb genomic segment on the genome replaced with a synthetic segment. And one important design feature of the Rexer system is the product of the first round of Rexer will serve as a template for the second round of Rexer right away. And basically, in the second round of Rexer, you basically synthesize the consecutive second 100 kb of synthetic sequence designed to replace the consecutive second 100 kb on the wild-type genome. And then you select for the gain of +1, which is now coupled with the synthetic DNA, and the loss of -2, which is now on the genome. Right? Then in this way, you would have the second consecutive 100 kb on the genome replaced with purely synthetic sequence. Basically, everything genotypes and phenotypes correctly. And with this ability, and also with the ability of using the bacteria artificial chromosome, which allow us to hold at least 300 kb of synthetic DNA, we actually envision we could basically convert the entire wild-type genome, which is running the size of 4.5 kb, 4.5 megabase, into a brand new, de novo synthesized genome in around 15 steps. And each of the Rexer steps takes around a week. So basically, if we have the budget to actually really pay for the synthetic genome, potentially that genome can be made in like a few months. Right now, basically, we have also another thing. Basically, we call this strategy genome stepwise interchange synthesis, or shortened as Genesis. Now, basically, with the new strategy of Genesis, we have the ability to actually convert the wild-type genome into a synthetic genome. Then the next question is, how do we actually design the synthetic genome to actually achieve our goal of expanding the decoding space of the natural life system? Right. Basically, the idea is basically if we focus again, for example, on the 6-fold degeneration decoding box of serine, we first identify the codons we are target for genome-wide removal. Then we are trying to remove them, but replace them with their synonymous codons. And if we could achieve that, then basically in that intermediate genome, all the TCG, TCA target codons would be completely gone, such that their cognate tRNAs will not be redundant. Then we can delete these redundant tRNAs. Then basically we completely freeze up the decoding space as for the TCG and TCA. Then we can basically reassign them to encode for unnatural amino acids, right? Then basically the question is, how do we actually determine which one of the remaining synonymous codons we can use to actually replace our target codons for the genome-wide removal? We cannot do this randomly because codon choices matters, and we cannot do this combinatorially throughout the whole genome because the number of the genome required to actually explore all the possibilities exceed the number of items in the universe. So basically, the only way we envision to actually answer this challenge is we're trying to identify the best synonymous replacements by matching codon-specific decoding properties, and also ideally trying to basically validate on a defined region before we roll out onto the whole genome. And basically, we try, for example, these are the TCG and TCA target codons. We are trying to identify their best synonymous replacement by matching codon-specific translation efficiencies. In this case, measured by different indices specific for codon-specific translational properties like CAI, codon adaptation index, TAI, and TE, which is a third indices we actually developed in-house. And by implying like these different indices, we actually identify the best potential targets. Then basically, we also did the same for the leucine target codons and alanine target codons. Then basically, we also identified a 20 kb essential region, which is a cell division operon, which has the highest density of such target codons across the whole genome. And for a few other details I wouldn't go into, basically this leads us to believe this 20 kb particular operon is the most challenging 20 kb operon in the 20 kb region across the whole genome to actually recode. Then basically, we synthesized different synthetic 20 kbs based on the different recoding schemes. And then the plan is we try to use the synthetic 20 kb following different recoding schemes to use REXA to attempt to replace the genomic 20 kb sequences. And if we are successful, then that is an indication like these particular recoding schemes would be viable. Potentially, they can be good candidate for us to roll out onto the genome-wide recoding. And we first actually tried with serine recoding schemes. In particular, we tried the serine recoding schemes 1, in which TCG and TCA are recoded into AGT. And when we tried this, we are actually initially very confused because for all the clones we actually picked up from after the REXA, although the beginning and the end of the 20 kb are always 100% integrated into the genome, in the middle of the 20 kb, we always see a chimera. And basically, let me walk you through over here. And these red lines indicate the position of the affected codons. And if at one given position, if the recoding is successful, we give that position number, the value 1, and color that position right. If that position remains wild type despite the attempt of the REXA recoding strategies, we give that position value 0 and color that position black. Then basically, for the recoding scheme 1, all the survival clones show the chimera across the 20 kb. And then what we did is that we overlay all these individual recoding sequences into something we call the compiled recoding landscape, which indicates to us there's just one single codon position across the entire 20 kb that always remains as wild type, can never be recoded in our attempts. Then we think this is really confusing and we don't know what's going on. Then we tried different recoding schemes. Recoding scheme 3, which is highly related to recoding scheme 1, which is only differed by a single nucleotide in the recoding rules. However, for recoding scheme 3, basically all the recoding attempts just works all the way through. Essentially, for the majority of the clones, they have like full recoding from the beginning to the end. Then we were very confused. Then we were kind of wondering what's going on here at all. And this is what we think is going on. And potentially, if we have a viable recoding scheme, for example, in the case of recoding scheme 3, then the synthetic 20 kb, the synthetic DNA built based on this particular recoding scheme is also viable. Then basically, in such circumstances, we can just replace the genomic fragment with the synthetic fragment straight away, leading to basically like straightforward replacement of the entire 20 kbs. However, if a particular recoding scheme, like in this case, recoding scheme 1, that is not fully viable, then basically the 20 kb synthetic DNA we generate based on the recoding scheme will contain synthetic lethals. Then basically halfway through the REXA pathway, we would actually generate an intermediate genome which actually contains the synthetic lethal on that genome, which is not viable, which will kill the cell. And now that actually we are only selecting for the survival of the colony, the only way for the cell to actually survive this synthetic lethal is to use the wild type copy of the fragment, which still hangs on for a little bit, to actually cross over to replace the synthetic lethals. And then basically that we basically expect is the mechanism that's giving us all these chimeras, which we see with the recoding scheme 1. And taking advantage that we have tremendously high efficiency using the REXA systems, we have like millions of colonies. We can overlay all these different chimeras, which will give us the compiled recoding landscape, which will not only basically allow us to differentiate viable recoding schemes from the non-viable recoding schemes, but more importantly, pinpoint precisely where exactly the non-viable recoding schemes this is synthetic or lethal is, which is in this particular codon position, which is the 407th codon position in the FTSA genes. Then the question is, and basically here basically shows this codon position. And for the other two recording schemes, it basically works all the way through. Then the question is, can we actually fix this particular identified synthetic lethal? And the answer is yes. If we basically replace this lethal AGT codon with alternative replacement, AGC, which has been proven to be working in recording scheme 2 and 3, then basically just by changing that single nucleotide in that single codon, then basically without touching anything else on that 20 kb region, we fully revive the previous completely lethal recording scheme 1. Now basically works all the way through. So basically now, we not based on the Rexer systems, we not only have a method to allow us to really pinpoint such design flaws on the new genome, but also allow us to very efficiently actually fix such design flaws. And essentially, this really gives us ability to actually walk around this cycle of design test, identify flaws and fix flaws very, very quickly, which not only basically allow us to fix all the potential design challenges as we on our way of making the de novo synthesized, fully synthetic genome, also basically lead us to find out all these hidden features on the genome design, which will deepen our understanding on the genome structure. Yeah, we'll be done in a minute. Right, then basically all these, and these are the basically all the remaining recording schemes. Basically, we did everything. And for the recording scheme 7 and 8, which are the alanines, they only differ again by a single nucleotide, but one works all the way through, replacing almost 400 codons at one attempt, while the other one fails catastrophically. And these are all the different recording schemes. And essentially, these three works straight away, while this particular one requires one single nucleotide fix. But once you fix them, there's no growth defect at all when compared to the wild type strain. And basically now we not only have, we truly have actually a way to actually make a new genome. We have the way to actually design the new genome. We also, more importantly, have the ability to actually debug our potential first version of the design. And what we would like to do in the near future is really put the notion into action and really make a new synthetic genome to support synthetic life that fundamentally go beyond the limits of nature. And that is the end of the talk. And I would like to thank all the colleagues, all the comrades, which we all work together. Julia single-handedly mastered the yeast assembly strategy to basically assemble all these long pieces of synthetic DNA in yeast. And Simon and I basically developed the bioinformatics strategies for the different recoding schemes. And Sam and Tian-San basically helped for doing the experiment. And all this, I have to say, has been supported by Jason. Essentially, all this idea essentially started as one elusive dream on a piece of paper. And Jason really supported us to basically pursue this apparently seemingly mad idea. And thank you, everybody, for the talk. Sorry.
We'll take questions after the session in the interest of time. Our final speaker for this session is David.
Our next speaker is Kevin Esvelt, professor at MIT Media Lab. Kevin is known for his seminal work on phage-assisted continuous evolution, known as PACE, as well as his work on gene drives for ecological engineering, which he will be discussing today.
Thank you for the kind invitation and to the organizers for putting everything together. So let's talk something that is potentially even more controversial than GMOs in foods. And let's also see if we can get a question to get even more people to raise their hands. How many of you have engineered an organism? Please raise your hand. How many of you have worried that were your organism to escape, that your engineered allele would outcompete the wild type version? Wow, okay, this is actually highly encouraging. Normally, we don't worry about that, of course, because when we're engineering an organism, we're doing it for our benefit, not that of the organism. And in general, we're diverting vital resources away from survival and reproduction to doing whatever we want. And that's especially true in the ancestral habitat. It's one thing to put in herbicide resistance, and then that plant will have a selective advantage on a farm where we're spraying herbicide. It's another thing entirely to have it spread in the wild. But of course, now with modern genome editing, we can in fact do this, or at least genome engineering. How many of you have delivered into a cell a single piece of DNA encoding both the CRISPR nuclease and guide RNAs that target the genome? Oh, that's not so encouraging. All right, hopefully you're doing something like viral defense. But even there, I hope you're not doing it targeting the genome, because of course, if you do this, deliver in both your nuclease and your guide RNA, along with whatever change you want to spread, and your guide RNAs target the genome, then of course, genome insertion will happen per normal for CRISPR. But once you get this in, then you have a functional copy of CRISPR in the genome. It can then cut and edit the other copy. That is, this is one way of getting a homozygote in a single step. But that is not, hopefully, why you would do this. And hopefully you wouldn't do this for any other reason. You would do it because of what happens when this organism mates with a wild-type organism. That is, all the offspring are guaranteed to inherit at least one copy from the homozygous parent. And in the offspring, genome editing happens again. You convert heterozygotes to homozygotes. So when they mate, all of their offspring will inherit your desired trait and the CRISPR system, and on down through the generations, until in principle, if you designed it correctly, you could edit every organism of the target species. This is not wise. And the real reason is, of course, as we all know, when we're engineering any biological system, it's very complex. We don't understand what all the parts do. And things go wrong. It just happens. Experiments fail, especially in biology. And that means that, in general, we should always be trying to make the smallest possible change that can solve the problem we're interested in. And we should start small and scale up if warranted. So in the case of environmental changes, agriculture and the like, this is why we run field trials. Make the change in the local environment and see what happens. But this is self-scaling. How self-scaling is it? Well, if you run a simulation of a weak CRISPR gene drive system, so this is actually the least effective CRISPR gene drive system described in the literature to date. This is in fruit flies. It has a copying rate, that is, it goes through homology-directed repair rather than non-homologous end joining, only half the time. And the rest of the time, it makes a drive-resistant allele that blocks its spread. Middling low fitness cost of 10%, but no cost of resistance. So these drive-resistant alleles are just going to pile up and block its spread. There, simulations show that it still goes up to more than 30% of the local population before it gets outcompeted by those drive-resistant alleles. 30% of the local population is more than enough that any level of gene flow with neighboring populations means your drive system is going to spread into that neighboring population. And from there, it's going to go up to 30% and probably spread into the next one, and the next, and the next, and the next. So here is how you could get eventual spread to every population, even though you're almost certainly not having any ecological effect, just because it never becomes abundant enough in any one population. How many organisms are required to do this? Well, if you run these simulations and you assume no bizarre incompatibility effects between your lab strain and the wild strain, the answer is, well, it can happen with one. This is the maximum this is the frequency distribution, the maximum frequency attained, and you can see the sort of the shaded yellow is the probabilities. And by the time you're up here beyond 10 organisms released, it's going to go. Is this a problem ecologically? Probably not. Most changes that we make are pretty unlikely to have any major ecological effect. Again, it depends entirely on the change, total case-by-case basis, but probably that's not really what we're worried about. We're also not super concerned about biosecurity. And the reason for that one is these things spread over generations. They're slow. You can detect them. Just sequence anything, and if you see a single read that has a eukaryotic expression signal and a CRISPR component in the same read, that's unnatural. And you can't build a gene drive system like this without one. So anything that's slow, easily detectable, and of course can be countered just by building another one, which we've shown works, is not much of a weapon. So we're not worried about that. Why are we concerned? Well, we all do genome editing. Some of us work in more controversial fields than others with respect to genome editing, but we should all be very concerned about whether there is truly public support for what we do. And the reality is that this is too invasive to be practical. Drive systems are extremely likely to spread beyond field trial sites, meaning you can't run that field trial. And the more people and nations that would be affected by any proposed intervention, even malaria, the harder it will be to reach agreement. In the time I've been talking, malaria has killed probably 8 children. And even there, how difficult is it going to be for all of the African countries that harbor the responsible mosquitoes to agree to release a gene drive system to block transmission? That's an unprecedented level of international agreement, especially concerning something as controversial as genome editing of wild populations. And of course, a single laboratory accident could put paid to the long-standing dream of ending malaria by disrupting the transmission through the vector. Again, 10 organisms, and the odds are it's going to take off. And you can imagine the headlines. I don't know how many of you have stock in Editas or Intellia or anything like that, but if you do, you really don't want to see a headline that says, scientists accidentally convert entire wild species to GMOs. Is CRISPR to blame? We really don't want that. And if, notably, if that delayed target malaria's efforts by, say, a decade, then that would result in 4 million children dying that could otherwise have been prevented. So when you're next delivering DNA into an organism, especially if it's a fruit fly, please be careful. That's why, in fact, we've been really emphasizing this. We need to be careful about this technology. From first describing it before we actually showed it that it worked in the lab, calling for regulation, developing in the first examples in yeast, demonstrating that all of the safeguards that we proposed worked, and then gathering a group of other scientists to agree on what the recommendations for laboratory research should be. We're trying to prevent that laboratory accident that would just devastate the field and even associated fields. But if we want to actually do something useful, other than trying to persuade countries to go ahead and get rid of malaria and schistosomiasis, we really need some way of reducing the number of people that have to agree. In other words, we need a way of confining the effects to a small region so we can safely run a field trial and only need approval from one national government at most. So we need a local drive system. How do you do that? Well, one of the confinement mechanisms we proposed said, okay, the problem here is that it has everything it needs to spread, so let's just separate it. That is, if you build a drive system that is just guide RNAs and in a host that is CRISPR positive, it will work, but it will only work in hosts that are CRISPR positive. So if you release these, then you would have a bunch of these. You'd have your B alleles, but they don't drive. And they cause A to drive, so it increases every generation. But it's really just too slow to be useful. If instead, you separate it into 3 elements, a serial chain, we call a daisy chain, then C drives B, B drives A, and now you're starting to get something, because the B to A does result in an increase in the population. Now C to B does, but as B increases, over time, A increases even more. So you get a much more powerful amplification effect in the local population. But of course, natural selection is still here. It hasn't gone away. It's dragging down the frequency of all these alleles, which means the net effect is kind of like a multi-stage genetic booster rocket. The more of these daisy drive alleles you add to the chain, then the more powerful it is, the greater the spread you'll get of your A element. And to give you an idea of the comparative power of these, obviously it depends on the fitness cost and homing rate, but here's your 2-element split. Here's your split drive, 3-element, 4-element, 5-element. And of course, if we go out even further to say 7 elements, then 90% homing rate, you have 7 elements. The most relevant plot here is this one. So what is the frequency you need to release in the local population? How many organisms per population size you want to modify in order to get the frequency to essentially fixation? So 20 generations, I mean, depending on the organism, you probably don't want more than that. So if you follow the 20 line out, you see you need to release for a 5 or 6 or 7 element daisy drive, you need to release about 1 in 50. But the best drive systems in mosquitoes to date have found that the homing rate in the germline of the mosquito is actually 98%. And if you do that, now you're out here in territory of 1 organism per 500 that you want to edit. So this is way more efficient. For comparison, the sterile insect techniques that have been used against mosquitoes, primarily in Brazil and on various islands, and not so much in the Florida Keys, they release 5 sterile males per 1 wild mosquito. So we could be, in principle, 3 orders of magnitude more efficient. But there's another way to do this. Instead of chaining them together linearly, you can just have lots of B elements. So if the first one was termed a daisy chain drive, this is a daisy field. You just have a field of guide RNAs, in this case, driving the A allele that has your CRISPR nuclease and whatever your desired change might be, potentially. So daisy field, say you have just 4 guide RNAs scattered everywhere in the genome that in turn drive the CRISPR gene and whatever cargo you want, make it homozygous. When you have 4 copies of these guides in your initial organisms, they mate with wild type. On average, the offspring are going to inherit 2. And then it's going to be down to 1 and then 0. So you can see the drive runs out of steam. But it still does spread to an appreciable extent. So now we're looking at ways of inserting lots of copies of guide RNAs throughout the genome to quickly create a powerful daisy field drive system that still won't spread indefinitely. So daisy chain, again, you just serially link the elements together where each drives the next. Versus daisy field, you simply have lots of copies of your B element, all of which drive A directly. And they have different strengths and weaknesses. But none of this solves the problem of gene flow across political boundaries. So one of our other projects that we're not actually using any kind of drive system on, we're trying to prevent Lyme disease and other tick-borne diseases by immunizing the mice that infect most ticks. So suppose this works. Suppose Cambridge decided that they wanted to immunize all of the mice in Cambridge. But what if Somerville said no? It's not like we can tell the mice, no, don't wander across that boundary line. Mice don't know that it's there. So how can we do this? We need some way for the genes themselves to tell whether or not they're supposed to be there, and for natural selection to act accordingly. So what we need is we need them to call a quorum vote. If there's a lot of engineered genes in an area, then they're probably supposed to be there, and so they should be favored over the wild type. But if there's only a few, then they're probably not supposed to be there, and we want the wild type to outcompete them. So how do you do that? Well, you can create a genetic quorum by working with haploinsufficient genes. So most ribosomal genes, you need 2 copies for the organism to survive. If you identify 2 of them and you swap their positions, then you end up with a heterozygote. It has 2 swapped copies. When it mates with a wild type organism, 1/4 of offspring inherit 2 swapped copies and are fine. 1/4 inherit 2 wild type copies and are fine. But the other half inherit 1 of each, and therefore are imbalanced, and so they die. In other words, it's really bad to be a hybrid. And in an environment where it's really bad to be a hybrid, the phenomenon is called underdominance, whichever allele is in the majority is going to win, because it's more likely to mate with something like itself, and then its offspring will not be hybrids, and they will be more fit. The neat thing about this swap is that it's something that we could spread with a daisy drive. either daisy chain or daisy field. If you have a daisy element in the form of guide RNAs here, and here are your haploinsufficient genes, we swap the positions. Oh, that's an interesting effect. See, this is, when this sort of thing happens, it makes you wonder, should we really be engineering biology, especially out there? Like, really? Oh, okay. Well, let's look at these ones down here then. Whoa. Here you have a daisy element, and you have a CRISPR nuclease next to your swapped ribosomal genes. The daisy element catalyzes the exchange. It cuts the wild type brown, replaces it with recoded green and CRISPR nuclease, cuts green, replaces it with recoded brown and CRISPR nuclease. In other words, all the progeny are guaranteed to inherit 2 swapped copies as long as this daisy element is here. But only half of the progeny are going to inherit the daisy element. Once you run out of daisy elements of one form or another, you're left with hybrids. And again, it's bad to be a hybrid. So the net effect is you could release daisy quorum drive organisms into Cambridge, and then we could monitor how far they spread, release more here, more wild types over in Somerville if necessary to push them back. And where the drive would cause it to spread out towards the edges, and when we get close to the city boundary, it runs out of daisy elements, and then it starts making hybrids, and the hybrids are less fit. And so in Cambridge, you'd have selection for the engineered variants. In Somerville, you'd have selection for the wild variants. That is, any engineered mice that wandered across the border would mate with wild types, create hybrids that would be disfavored. Natural selection would wipe out the engineered alleles. It's not perfect. We would love to have an absolute barrier, but it's much better than what we have now, and it might even be enough. Who knows? But most exciting, we have to face the fact that CRISPR is becoming more and more accessible. There are even high schools now that use CRISPR for genome editing experiments. And while building an actual gene drive system is, of course, much, much more difficult than engineering some E. coli or yeast, the limiting step is the transgenesis. There are also many well-funded high school labs that work with fruit flies. I don't know if any of them do transgenesis, but conceivably they could. At the very least, there's thousands and thousands of people that can do it. And that number is probably only going to grow. After all, we can all see the incredible potential of genome editing. So, sooner or later, it is highly likely that someone is going to edit the environment without the consent of the affected community. We desperately need a way of restoring that population to wild type. So how do you do it? Well, daisy restoration drive is basically that daisy quorum drive I just described that's engineered to overwrite its target. But what we really need is a way of overwriting and replacing every copy of a rogue invasive CRISPR gene drive, the kind that spreads forever. So how can you do that? Well, if you take a daisy quorum drive and you add some guide RNAs that piggyback on the nuclease of the rogue drive system, and your daisy quorum uses an orthogonal nuclease, then in the presence of the rogue drive nuclease, it will spread like an invasive CRISPR drive system. It will spread indefinitely, and it will spread all of the daisy elements. It will drive even the daisy elements, not as well as your actual daisy drive nuclease. So if you parachute in a few mice, assuming it's mice that have been edited here, into the area that is where you've detected the unwanted rogue drive system, then your daisy drive will begin spreading like an invasive drive system until it overwrites every copy of the invasive drive system. But in order to do that, you can't allow the invasive drive system to continue spreading through wild type. You can't just play catch up. It will always have more wild type nearby. But with this one, when it hits the wild type population, it still keeps copying and spreading through the wild type and immunizing them against the rogue drive system. The difference is it relies on its daisy elements to do that, and so it starts losing them. So in other words, it spreads efficiently and overwrites every copy, then spreads a little bit, immunizes the nearby wild type population, so it has a chance of getting every copy of that unwanted drive system. And then you have this daisy quorum effect on the edges in particular. It's bad to be a hybrid. So if we just release a lot of wild types, or better yet, since all of these have CRISPR nucleases, your daisy nuclease, if you drop in a suppression drive element, which is just guide RNAs that target, say, a recessive female fertility gene, so that any female offspring of 2 parents who are carriers are born infertile, then you can selectively suppress the engineered population as soon as it's in the minority, then it's at ever more of a disadvantage due to the quorum effect. All the engineered changes are linked to the quorum effect, and so population becomes wild type. Is this Rube Goldberg? Well, yes, it's Rube Goldberg. It's totally Rube Goldberg. But it's Rube Goldberg where every last step is CRISPR genome editing. So any other biological system, I would say there is no way we have a chance of getting this working. But because it's CRISPR, and CRISPR is frankly magic, I think this has a good chance of providing a way to ensure that the shared environmental commons is whatever the people who live there want it to be. That does not allow single actors to unilaterally decide what the environment should be like. And perhaps even more excitingly, this isn't limited to invasive rogue drive systems. This is something that we could use to potentially restore the wild type any population with any engineered genes at all, whether they leaked from some other species that we meant to engineer or whatever other reason. If it's drive amenable, we could potentially restore it to wild type using a method like this. And more broadly, this ability to edit wild populations could do a lot of good in the world. And not just for humans, although there are certainly many health applications, from mosquito-borne diseases, tick-borne diseases, to parasites that are sexually reproducing themselves and could be targeted directly, to even animal reservoirs of disease. What about animal welfare even? I mean, right now there's a lot of kittens that starve and freeze in the wild out there. And we spend a lot of money adopting them, but there's always more. Wouldn't it be lovely if we could ensure that every kitten was born, came into the world destined for a warm and welcoming home? Wouldn't it be lovely if we could, say, remove the invasive mosquitoes and rats that are driving species extinct on islands? And I could also say, wouldn't it be lovely if we didn't need to spray our crops with herbicides and pesticides in general and instead just engineered the pests to suppress the local population? Or better yet, tweak them so they just didn't like the taste. But if we're going to do that, frankly, I think this needs to come last. These need to come first, because priority is tackling problems that have obvious benefits to the typical person on the street. And again, since I began talking, we're probably up to 16 or 20 children that have died of malaria. So problems like that have to come first. But more broadly, this is a technology that can engineer the shared environment. And that means that if you develop this behind closed doors, you're directly denying people a voice in a decision that could affect them. And I think we need to use this as a test case for a new way of doing science. That is, one where we do pre-register our proposals so that everyone knows what the field is at least attempting to do. And that means, because based on the few reactions of a few of you, who are probably not so keen on sharing your best ideas so they can be scooped by some other laboratory who has more money in hands, we need to change the incentives governing science, at least for this field. And there are a number of ways we can do that we're looking into, from the journals to the funders to even intellectual property. I mean, the Broad took a powerful step by denying Monsanto the right to use CRISPR for gene drive explicitly in that licensing contract, as well as for Terminator technology and other things. So IP ethical licensing is a potentially valid tool that we could use to change the incentives governing particular fields of science. And this is the field where we need it most. We've gone ahead and pre-registered all of our experiments involving any form of gene drive on bioRxiv, and more recently on a responsive science site. And we're trying to adopt an approach where we work with communities to decide what exactly they need so that they can help guide the technology from the very beginning. And this is in large part because I don't trust myself. Technology is becoming so powerful, and it's so difficult to really apprehend the possible consequences, especially when you combine it with advances in other fields. I mean, look here, CRISPR-based gene drive. Austin Burt, who is an absolute genius at Imperial College, who's been leading the target malaria effort, first proposed that we could use homing endonucleases like ISKI1 from yeast to edit wild populations using exactly this form of gene drive in 2003. But he never imagined that we would have CRISPR and that we could therefore potentially apply this to many different species so long as they're sexually reproducing. It's hard to apprehend consequences. And so by openly sharing our proposals before experiments begin, we give the rest of the scientific community a chance to not only decide what they want to do in an open and rational manner. Do you want to intelligently collaborate or intelligently compete, as opposed to the blind paranoia that characterizes most of the CRISPR field? Do you want to, we can actively invite concerns from both the rest of the community and also from local communities of citizens who might be affected. We can actually request guidance from the sites that might be candidates for early field trials and ask the people there what they would like to see. Of course, arrange for independent assessment of any of those field trials, because just like the last person you want to test a hypothesis is the person who proposed it, same goes for evaluating a technology. And lastly, we can, above all else, start local and only scale up if warranted. Thanks in particular to all my students and collaborators that have worked with me over the last few years on this, various funders, but especially to my lab, who have really gone out on a limb. They're the real heroes of this story because they are willing to share their best ideas and, at the moment, risk somebody else scooping them. So thank you.
So we can take a few questions.
In this scenario, like the quorum scenario, does this essentially require people continuously monitoring this and adding or taking away something from the system, or will this become a self-sustaining thing?
Great question. You definitely need to monitor in order to know what's there. So just because you can unfailingly detect a drive system present in a given species, I mean, my hope is that advances in sequencing will eventually allow us to simply sequence the river at the bottom of every watershed and detect any drive system present in any species that lives in that geographic region. Worst case scenario, we have to sequence the populations that we're concerned about. And that's not something that is a priority for the next 5 or 10 years, but eventually it's the sort of thing that, well, let's just say that ecologists should rejoice because they're going to get on the Department of Defense gravy train.
Are you concerned that the restoration through a gene drive would eliminate the diversity in the wild type?
Yeah, so should we be concerned about gene drives removing diversity in the wild type? So mostly what the drive system does is it changes the one gene that you're targeting, or some combination of genes. It's possible that if you have an off-target, that's most likely to just lead to a fitness cost. But you can imagine that an off-target might create a mutation somewhere else in the genome and then drive it through the population. But for the most part, you're not affecting the vast majority of the genetics. You're just affecting those that you intend to. So hopefully you wouldn't do it at all unless you're sure that altering at least that allele in that particular local population is a good thing, and ideally through local field trials to test that out first. More broadly, that is one reason, very excellent reason to be concerned about the self-sustaining invasive version that spreads indefinitely, because there you probably are going to affect every organism of that species. Yeah.
So playing devil's advocate here, you could say that in the 1970s, Cambridge was shit scared of the common DNA technology. And so how do you contract those kinds of arguments that this is something that will not really be as bad as it can be?
Right, so how do we, given the moratorium from the 70s on recombinant DNA that Cambridge imposed, how can we ensure that that doesn't happen? Well, I'm just
saying that maybe it's not as big a concern.
So again, we now know that we don't really have to worry about SV40, recombinant SV40 spreading through populations and causing plagues. And we know that recombinant DNA is not going to break evolution. This doesn't break evolution. Gene drives are natural. They occur in just about every species on the planet, including us, because a transposon is just a different form of gene drive system that spreads even though it does not benefit individual organisms or the rest of the genome. So it's not really anything profoundly new and different. I don't think it's particularly threatening at all. It's not a good weapon, and most changes are not going to have ecological effects. Even if they do, they're going to spread slowly, and hopefully we can perfectly reverse them as needed. Now, it's true that with an ecosystem, if you build a dam, and then you knock the dam down, it's not necessarily going to go back to the way it was before. But that's true of anything, right? That's true of anything at all. So I'm not concerned by anything other than the risk to public perception, because we are, to some extent, in a hole when it comes to the public trusting us. And in many areas, frankly, I will give this to, say, Friends of the Earth and ETC group, whom I disagree with on almost everything, but they do have some valid concerns about the way that we do science. I think that by being more open and responsive and actively reaching out to communities, saying, please help keep an eye on us, help think of things that we might miss. You're the ones that live there. You know more about the environment. Figure out what might go wrong, and then we can hopefully address it in the early stages of technology development before it happens. And then there is no substitute for good communication with the local community. In about 3 hours, I'll be addressing the Cambridge Biosafety Forum and training the IBCs for local biotech companies and universities. So doing that kind of local outreach and communication is absolutely essential.
How has the response been to the Oxyca's experiments in limiting and all that? Has that been received positively by political groups in other countries?
Oh, so Oxytec? Yeah, so Oxytec makes the sterile male mosquitoes. And those have been received incredibly positively in, say, Brazil, where there is a lot of dengue. And the people say, wait, you can do something about mosquitoes so we don't get dengue? That's fantastic. In areas like the Florida Keys, you had a recent vote as to whether or not the trial should go forward in Key Haven. The citizens of Key Haven said, no, it should not go forward. The citizens of the rest of the county said, yes, it should go forward in Key Haven. So in that case, what they're going to do is they're picking another section of the county that had voted yes to Key Haven, and they're going to release it there without asking them again whether they wanted it. So this is the NIMBY effect. We know that it is real. But at the same time, I don't think they're doing anybody any favors by moving ahead in a community that said, yes, but only over there. I think the Keys have not had a dengue outbreak for 7 years. And even then, it was just a couple of people. They just don't see it as a large enough problem to take this step, and there have been a lot of missteps by the company in reaching out to the community from an early enough stage. I mean, we have this idea as scientists that if we explain to people what we're doing, then of course they'll support it. Because what we're used to is we say, okay, give me all the available evidence, and I will look at it, and then come to a conclusion about what I think is right. But that's an appallingly hubristic attitude that assumes a great deal of expertise in the relevant area. That's just not how normal people think. We're sort of brainwashed into thinking that way. Normal people just want to know, so first of all, who are you proposing this? Why do you want to do it? Who's it going to help? And what's it going to cost? And who's going to pay the bill? That's what they want to know. And that's a much more reasonable set of questions, assuming that somebody else has the relevant expertise, wanting to know that whoever that is has access to all the data. I mean, this emphasis on transparency and openness, it's not that we expect the vast majority of citizens to even go and look and see if it's there. They just want to know that in the event that someone who does have the relevant expertise takes an interest, then they will be able to see everything that, say, the regulators are seeing. And above all else, I cannot stress this enough, we absolutely have to stop relying on the regulators as a proxy for community approval. Because the FDA only invites comments at the just prior to product approval, when it's far too late to actually change anything about it. And then they invite comments from the public. 90% of them are questions of ethics and values. And the FDA throws all those out because they say, well, you know, our mandate is only to evaluate empirical data and likely outcomes. And so it's like they invite people with their opinion, and then they say, nope, these opinions, not relevant. That's just not good community outreach. So we need to do better as the scientists from very early on addressing these kinds of issues, because at the end of the day, we should hold ourselves morally responsible for all the consequences of what we do. And in a world where it's getting ever harder to predict those consequences, that means we need to invite other people for help.
Great, let's give another round of applause. So this concludes GE 5.0. Thank you all so much for coming. Without you and your enthusiasm, this conference wouldn't be possible. And also thank you to all the sponsors and all the speakers.